This is a walkthrough of the procedures for Exercise 2 of the Physical Properties and Concentration Lab. Step 1 is the same as always. Gather your materials. For this experiment, you'll need blue water and the 20% stock solution provided to you. A stock solution is a concentrated solution that we prepare to be diluted in our experiment. Make sure you transfer your solutions to a beaker instead of taking the container directly. The other materials you'll need for this experiment are a pipette and bulb and a 25 milliliter flask. Remember your scale and
don't forget your safety goggles and gloves.
Before we continue with the procedures, remember your safety instructions. The solutions in this experiment are mild. However, if some were to come in contact with your skin, remember to flush with water and wipe up the spill. In this experiment, excess solutions may be poured down the sink. Remember, when using unknown chemicals, use small quantities. Now that you have your safety instructions, you can begin the fun part of the experiment. Step 2 of your experiment is to weigh the empty 25 milliliter container. To ensure that your measurements are accurate, remember these few things when recording mass. Record the mass in your lab notebook. You'll need it for later calculations. The next step is to use the meniscus in the pipette to draw 10 milliliters of the stock solution. To make your data more accurate, make sure you read the bottom of the meniscus for your measurements. Now that that's done, pour the 10 milliliters of the stock solution into your empty 25 milliliter flask that you just weighed. Now head to your scale and take the mass of the 25 milliliter flask with the solution inside. Split up with your lab partners and allow one lab partner to do the calculations, while the other two repeat the trials to ensure that your data is accurate. Here are the calculations you should be performing at this part of the experiment. There are also a few examples provided from our procedures. Now you're done with about 1/3 of the experiment. Clean your materials and repeat the entire experiment. The only difference should be you're using 10 milliliters of the blue water instead of 10 milliliters of the 20% stock solution. Perform the same calculations as before, but with this new data. Again, some examples are provided from our experiment. Now we need to make a diluted version of our 20% stock solution. Diluting a substance means to make the substance a little less concentrated by adding a certain ratio of water to the solution. For example, for one of our dilutions, we did 2 milliliters of water to 10 milliliters of stock solution. Our group performed 3 dilutions total, with 3 trials of each dilution. Again, this is just to ensure accuracy and gather more data. Now, with your diluted substance, for the 3rd time, repeat the experiment, steps 2 through 5. Keep volume in mind because this will most likely change from 10 milliliters. Repeat calculations and add the calculation of concentration. Repeat the experiment as needed and make a chart in your lab notebook to keep track of all your data. Lastly, don't forget to clean up and dispose of your materials properly.